In this script, we will delve into the fascinating topic of muscle contraction, exploring its various aspects at the cellular and molecular level. It is essential to have a comprehensive understanding of muscle contraction, as it plays a critical role in our everyday movements and bodily functions. From the intricate interplay of proteins to the coordination of calcium release, we will explore the mechanisms that enable muscle contraction. So, let us begin the journey into the captivating world of muscle contraction. Muscle contraction is a complex process that involves the generation of force within muscle fibers. It allows us to perform tasks ranging from simple actions like blinking an eye to more strenuous activities such as running or lifting heavy objects. A muscle consists of individual muscle fibers, which further contain myofibrils, the contractile units responsible for muscle contraction. These myofibrils are composed of repeated functional units called sarcomeres, which give muscle its striated appearance. At the molecular level, muscle contraction relies on the interaction between actin and myosin filaments within the sarcomere. The thin actin filaments have binding sites for myosin, a motor protein that undergoes a series of conformational changes during contraction. This mechanism, known as the crossbridge cycle, involves the binding and detachment of myosin heads to actin, resulting in the sliding of actin filaments toward the center of the sarcomere. Before muscle contraction can occur, a signal from the nervous system must be transmitted to the muscle fibers. This process, known as excitation-contraction coupling, involves the propagation of an action potential along the muscle membrane, triggering the release of calcium ions from the sarcoplasmic reticulum. The binding of calcium to troponin, a regulatory protein found on the actin filaments, exposes the myosin binding sites, enabling the crossbridge formation and subsequent contraction. The regulation of muscle contraction is intricately controlled to ensure precise coordination and prevent uncontrolled muscle activity. Troponin complex, consisting of troponin C, troponin I, and troponin T, plays a vital role in this regulation. Troponin C binds calcium ions, while troponin I inhibits the interaction between actin and myosin. Troponin T anchors the troponin complex to the tropomyosin, another regulatory protein that covers the binding sites on actin in the absence of calcium. Different types of muscle fibers exhibit variations in their contractile properties, which are attributed to the presence of different myosin isoforms. Myosin isoforms play a crucial role in determining the speed and efficiency of muscle contraction. Additionally, motor proteins such as dynein and kinesin facilitate the transport of cellular components, contributing to the overall functioning of muscle cells. Cardiac muscle is a specialized type of muscle found in the heart. It displays unique characteristics that allow for coordinated and rhythmic contractions, essential for pumping blood throughout the body. The cardiac muscle cells, known as cardiomyocytes, possess intercalated discs that facilitate rapid electrical conduction, ensuring synchronized muscle contraction. Calcium influx through voltage-gated channels plays a critical role in initiating cardiac muscle contraction. In skeletal muscle, the release of calcium ions from the sarcoplasmic reticulum is mediated by ryanodine receptors. These receptors respond to membrane depolarization and facilitate the rapid release of calcium into the cytosol. The precise control of calcium release is crucial for regulating the duration and strength of muscle contraction. Smooth muscle forms the walls of various organs, including blood vessels, the gastrointestinal tract, and the respiratory system. Unlike skeletal muscle, smooth muscle contraction is largely involuntary and is controlled by the autonomic nervous system. The mechanism of smooth muscle contraction involves the phosphorylation of myosin by the enzyme myosin light chain kinase, which promotes actin-myosin interaction and subsequent contraction. The sarcoplasmic reticulum is a specialized network of membranous structures within muscle cells that stores and regulates calcium ions. It plays a crucial role in ensuring the availability of calcium ions for muscle contraction. The sarcoplasmic reticulum takes part in excitation-contraction coupling and actively pumps calcium back into its stores during relaxation. The crossbridge cycle is a fundamental mechanism underlying muscle contraction. It involves a series of steps, including myosin binding to actin, the power stroke, and the detachment of myosin from actin. ATP hydrolysis provides the necessary energy for the crossbridge cycle to occur, enabling repeated cycles of contraction and relaxation. During muscle contraction, the sarcomeres undergo a change in length and tension. The elasticity of sarcomeres allows for proper function and enables the muscle to return to its original length after stretching or contracting. Proteins like titin, also known as connectin, play a vital role in maintaining sarcomere elasticity. Apart from troponin, calcium-binding proteins, including comedulin, are crucial regulators of muscle contraction. Comedulin binds calcium and undergoes a conformational change, leading to the activation of various enzymes and proteins involved in muscle contraction. Its role is particularly significant in smooth muscle cells. In conclusion, muscle contraction is an extraordinarily complex process that involves a delicate interplay of proteins, regulation, and calcium signaling. Our understanding of muscle contraction has improved over the years, revealing the intricate mechanisms that govern this fundamental physiological process. Further research in this field holds immense potential for gaining insights into various muscle disorders and developing therapeutic interventions to counteract muscle-related pathologies.